focus right now is on the immune system and going from that cellular biochemical networks to whole body level.
A nuclear war, or maybe it's an accident like happened in Fukushima some years ago, where you need to treat somebody. A lot of it is confidential, but on a high level, can you talk about how your computational biology work has applied to the research into drugs?
We had been talking about the application of the immune digital twin at the patient and clinician level, but the other application of these models is in drug discovery and development.
Welcome to the BioTalk Unzipped podcast, where we unzip the stories behind medical progress by sharing the latest and greatest advances in biopharmaceuticals and medical technologies in a fun, entertaining, and enlightening format. And now your co-hosts, Gregory Austin and Dr. Chad Briscoe.
Tom, so another area that I'm very curious about, because I did some of my PhD work when I did with Dave Hage at the University of Nebraska, was in modeling and simulation. So I'm curious, and this one's getting real pretty technical, but what sort of mathematical models are you using? Is it a lot of kinetic models for the interactions, and therefore a lot of differential equations you're modeling? Can you talk a little bit about the math? I don't want to make it too scary for some of our listeners, but I'm really curious, so I wanted to dive into.
Yeah, so depends on which process you're talking about.
Sure.
This is one of the challenges, is that you have different mathematical. There's no one size fits all when it comes to mathematical approaches and applying them to different biological processes. So signaling networks are typically, signaling pathways when you have a more limited system. And when you have the information about the different kinetic parameters, then you would use a set of ODEs and go with that. Now, with larger models, and so our approach is more comprehensive in that we believe we're working under the assumption that or hypothesis that these signaling networks are what allows the cell to process the information. And so you can't process the information if you look at just a single signaling pathway. And so we're trying to model as much as we can. Now that poses a challenge that, number one, we don't have the kinetic parameters across all these different pathways. Number two, that would be so many parameters in the end that set of equations just wouldn't be solvable. Yeah. So we're using more qualitative sort of approach. It's called logical models, where it's the mathematics behind it is Boolean logic. So a component is turned off and on based on these OR gates, AND gates, and NOTs, and different Boolean operators. And so in the end, so in a typical Boolean network, the output is 0 or 1. In our case, we made some modifications to it where it basically becomes a Markov process. And so it's in the end, the output is a probability that a particular signaling component will be active over a period of time. And so that's what we use for signaling networks. The metabolism that uses constraint-based modeling, linear programming. So you've got a bunch of linear equations, and then you solve those. Yeah. And then then you want to model at the cellular level. So we use agent-based models, and so each agent. In a typical only agent-based model, the agent would correspond to a cell, and then there would be some simple rules that tell you, okay, this cell is going to die after so many steps, or if it encounters these other variables in a particular configuration. But our agents, we stuff those logical models and the constraint-based models of metabolism inside of these agents. And that's what gives rise to the behavior of that agent. Okay. And then the extracellular environment is modeled with ODEs. And so we've got 4 different computational mathematical approaches, and the challenge had been, how do you wire these all together?
That was my next question.
and so yeah.
Yeah.
So we've done that, and it's working really well, and so that had been one of the challenges that we had to overcome to be able to make this happen.
Yeah, that's okay.
So
That's
some
super cool.
I'm kind of above my head, but baseline that's going to connect these different styles. How you get linear equations to talk to the Boolean logic that's happening with the signaling.
Yeah, so we go through different normalization steps to basically take the probability of that's the output of the Boolean network, and then to be able to convert it to a specific concentration and vice versa. So depends on which part of the which mathematical frameworks you're trying to wire together. They'll use a different sort of a approach.
Do you sort of build it at scale? And when I say at scale, I'm thinking, do you build it out and say, oh, and I don't know what the right number is. There's a billion, CD4 positive T cells in your body, and there's 500 million, I don't know, other cell B cells, or I have no idea what the numbers are. But do you try to build it somewhat to scale like that, or do you kind of scale it down and then you're going to build it up later?
We're gonna our first approach is gonna be through sampling. We won't be going. And the question is, if the distributions are appropriate, then sampling may be completely fine as the end result. If they're not, then we will have to scale it up to the actual numbers if it's possible, and that
Yeah, interesting. Because again, when I was doing this in the mid 2000s, just a simple, and it was we were just doing it on a simple desktop computer, the simulations, but it would take all night to run the program once, and so you kind of run it and you find out the next day. But then if you think about chromatography, where you might want to run a couple of thousand slices, I could only run like 10 or 20 because it was it took so long to get a response. So really fantastic.
Even in this case, I feel like it's relative, right? You look at experiments with mice. You run one experiment and it takes months to generate data.
Yeah.
And if the data is bad out there, the timeline, the time scale is in months. Here for simulations is at least in days.
Yeah. Wow. So you just have the computer churning away for months at a time.
Yeah.
Yeah, that's something. So Tom, I guess also, do you mostly work in whole, like thinking of a whole body system? Do you focus on certain organ systems, certain cells? You must be able to sort of break it down to look at different types of problems or different spaces, right?
Yeah, so our focus right now is on the immune system and going from that cellular biochemical networks to the whole body level. And so that physiology, basically a PK or a PBPK sort of layer that connects to the physiology. And right now, our compartments that represent different organs are limited to the circulatory system, lymph node, and the site of infection. Is our first use cases in the context of respiratory infection.
Okay.
And then we'll be adding more organ-specific models and compartments as the model is assembled. We can demonstrate first use cases, and then we have more resources to scale up or to expand it.
So Tom, you and I, of course, came to know each other through some collaboration that we're doing with University of Nebraska, the U.S. Army, Nebraska Strategic Research Institute. That research, I'll just give a short background since I'm involved in it as well. But that research is working with the Army, the Department of Defense, to identify and assess drugs for treatment of acute radiation syndrome. So when you think about a situation where someone's exposed to a nuclear or a radionuclide, so from either it could either be the potential for a nuclear war, or maybe it's an accident like happened in Fukushima some years ago where you need to treat somebody. but also our focus in the work we've done has been how do you treat somebody to protect them from a potential radiation exposure if, say, a soldier were going into war. So you think of the Ukraine, right? And going into the area around Chernobyl, if you had to fight a war, you'd want to be protected. Or if there were a potential for the use of a nuclear, of a nuclear warhead, God help us all that we never get there. But anyway, so that's a really quick high level for our listeners about what we've been collaborating with, and you've played a big part in that team. Not just providing brilliant insights and help and support thoughts, but you've had a real material impact on some of the work that we've been doing. A lot of it is confidential, but on a high level, can you talk about how your computational biology work has applied to the research into drugs, small molecule drugs, primarily for prophylactic use to prevent acute radiation syndrome?
Yeah, absolutely. So we had been talking about the application of the immune digital twin mostly at the patient and clinician level, but the other application of these models is in drug discovery and development, and in fact the whole spectrum from drug discovery all the way to clinical trials. In this particular project, we've been focused more on the drug discovery and then disease and characterization of the mechanism of action of that particular drug. So the disease and the drug, they impact these different parts of the biochemical process. And again, the biochemical process is a complex network, right? And so if you're using a drug to treat any disease, you're basically taking the drug, and that drug will likely shut down or decrease the activity of a specific signaling component or signaling components. Now, the question is, because it is a very complex network, a biochemical process, that drug is going to have a ripple effect through that network, and it's difficult to predict what other parts of the cell and then the human body, because the human body is a network of networks, as we discussed earlier.
Yeah.
How is it going to impact? And so then if you have a computer model, you can simulate that. If you have this complex model, you put in this virtually, you put in that drug, and then you can measure everything and see everything over time as it lights up or doesn't light up, as an effect in response to that disease or drug. And so our role has been building these models. We've been focusing on metabolism in this particular case to understand and then characterize how the disease may impact the different parts of the biochemical process, and then also if we apply this drug that's being tested, how will it impact other parts of the cells, so that we can better characterize and predict the side effects it may or may not have when the soldier actually takes the drug.
That's cool. And so that of course would be then that work should be applicable in the future to other disease states, and it's so
Absolutely.
You're developing a lot of these models for that. So that's really cool, Tom. Yeah, thank you.
All right, it's that time, Chad, for new news.
New news.
Yes, indeed. MIT's Sparrow redefines drug discovery with smart synthesis. So MIT Sparrow algorithm marks a leap forward in AI-driven drug discovery, helping scientists select the most promising molecules to synthesize by balancing the costs with the potential benefits. So Sparrow, which stands for Synthesis Planning and Rewards-Based Route Optimization Workflow, uses machine learning to evaluate and optimize molecular candidates for drug discovery by integrating predictions or properties, costs, and synthesis feasibility. This approach allows researchers to make informed choices on which compounds to test, accelerating the drug discovery process and improving efficiency in the pharmaceutical development. Sparrow has already shown success in real-world case studies, potentially setting a new standard for cost-effective data-driven molecular design. So How do you feel about this, Tom? What's your take, and how do you imagine maybe the integration of this technology with digital twinning?
Yeah, I think it along the same lines of what we discussed before, being able to use the technology to narrow down the possibilities that you want to pursue experimentally. Um, what is the cost to develop a drug now? Over like $2 billion.
Most drugs are certainly, well over 2 billion these days. Yeah. Yeah. Yeah.
And it takes over 10 years, and
Mm-hmm.
a lot of also the solutions are not personalized. And it depends on if you have what kind of a disease you have. If it's a rare disease, you may not be able to have clinical trials. Or acute radiation syndrome, you can't have clinical trials. You can't go and irradiate people to test the
Yeah.
medicine
Yeah.
on them. So
Right.
um, I think anything that can speed up the process and increase the efficiency and decrease the cost of drug development, I think holds a lot of value in my mind. And then again, combining that with digital twins that can be personalized. So plug in that molecule into immune digital twin, and then imagine, in the case of rare disease where you don't have enough population to
Mm-hmm.
to recruit to your clinical trials, but you have a computer model that you can create a virtual population from. So you can simulate much larger populations of those patients and test that compound on them. And so in diseases where the only way that you can go to a patient is through a non-human primate, and then you go straight to the patient, maybe there will be a room for a technology like that where you you can test on real humans, but once these virtual digital twins and virtual populations of patients are shown to be um, more accurate than NHPs, for example, and similar to running a actual human clinical trial, or even better, then I think I see a future where they become part of the drug discovery and development process as a necessary, required step.
Interesting. Do
So
you
it's interesting
foresee?
that, sorry, Greg.
Sorry.
Tom, it's interesting that, we've talked about a couple of different, applications of AI and whatnot outside of your digital twinning work. And then they both relate back. Do you have any collaborations going with any groups to do this sort of work and test out your, I guess we talked about the acute radiation syndrome work with the Department of Defense, but I was thinking with, more along these other lines of tying into other digital solutions. Let's call it that.
We have talked to a few people, and few entities, but we first need to build out
Right.
this multi-scale model,
Yeah.
because once we have that, then it's going to be a very easy conversation. Until then, it's too hypothetical, and
Sure. Yeah.
for us, it's been kind of heads down. Let's get this technology
Mm-hmm.
built to the state
Yeah.
where we can start developing specific use cases and
Yeah.
expand from there.
Cool.
Do you think that some of the future of digital twinning might also be used in recruiting clinical subjects for clinical trials based on, well, we think they're going to
Absolutely.
have the best effects? Yeah. Do you think if we do that, we get, these are the best, candidates, for our drug in the clinic, do you think that might skew the results a little bit? And so we've got the best conditions, and so we think, oh, it's 98% efficacious, and then it gets out to the regular population, and then it's not as great,
Mhm.
uh, as they thought it was.
Well,
Just depends on how you build the model.
yeah. So the way I hope that the future will be is that we develop these treatments for individuals because the way we do clinical trials today is problematic in a way that you still have a relatively small sample of population that you recruit, right? And in the case of the immune system, like we discussed earlier, everybody's immune system is just programmed very differently.
Mm-hmm.
And so we're developing treatments for sort of the average person, but none of us uniquely fit that average person's profile, and so I think in the future what we would want to do is develop these treatments that are specific at the individualized level, so that you can say, for you, the treatment regimen should be at this concentration, this many times a day or per week, and all of that. That would be, or you should be taking this drug in conjunction with a different one because it'll maximize the impact and minimize the side effects. And so that's where I think the ultimate future lies. In the meantime, I think what you described is sort of a middle bus stop where these models can identify potentially the subpopulations that would respond best for those drugs to those treatments. But there is also the opportunity from the other end of that, and that is that if you don't have enough folks recruited, or you could eliminate for a real clinical trial, you could narrow it down to the subpopulation that you feel would best respond to that treatment. At the same time, you can simulate then all the other subpopulations, so that you can understand what those other subpopulations would look like in terms of responding to those treatments.
Yeah, I can see, Greg, where right now we do that, right? We do optimize trials not to sort of skew the results, although that can be an unscrupulous way to do it. But we do it by race. We do bridging studies into Japanese populations who want to have a diversity of males and females. So there's all sorts of ways, fast metabolizers and slow metabolizers, and all these things that we have to do by genetic testing. And I've certainly gender and other things we can do other ways. But I can also envision then, as Tom was pointing out, where you optimize to a specific population that you wanna target, and so you can test that, or it can actually ensure more diversity in your trial, so that maybe run one trial that's bigger versus three trials that are half as big, and I can envision all sorts of different ways that you can re-optimize your trials. But I think everyone out there, including me, who makes my living off of running clinical trials, would agree that the ultimate goal would be that we don't have to run any, or we ran very, very few. Tom, that actually made me think of something. I think the great target is the human digital twin, but are you or anybody else, have you thought about building animal digital twins? Is that like clinical trials, we start in animals and build it up? Do we start in mice and dogs and build that up, or some other lower life form?
Yeah, I think absolutely there is the potential and need to do that. I think one of the we're not gonna, as much as we would like to get away from animal models, right? I don't think it'll happen anytime soon. But what we can do in the meantime is use these computer models to go from one species to the next in a more efficient way. And so having a digital twin of a mouse or NHP would enable us to go from one species and step of the biomedical research to the next in a more efficient way as well. In whether it's scaling the dosing or looking at the right pathways and the right organs as you move up the species ladder. I think that would be one of the utilities.
So it seems like you're heading towards, and I don't think it's going to be a single moment, but it seems like your research is heading towards a point where it's kind of linear. And then at a point you're going to say, we nailed this, boom. Now it's going everywhere, and it'll be, and then you'll do, where do we focus? How do we, and then there will be Tom Helikar students at different universities that are, picking up this work. And that's, very cool.
You're absolutely right. And our goal is to build it out as an infrastructure so that it's not just our, lab working on it, but make it available as an environment and infrastructure where other groups that, they're interested in, taking the mathematical and computational techniques we had developed and taking it from human to plants or different animal species and other systems.
Cool.
Do you foresee a massive database in the future of personalized anonymous digital twins that companies could then use? It's like, well, we want to test this particular drug, and they use real data from real people, though it's anonymized, and test in that kind of way. Sort of like, instead of, volunteering your body if you die to science, it's sort of, volunteering your digital body to science in that way.
That's a very good question. One of the ongoing discussions at different conferences that focus on digital twins, and including the federal agencies that we interact with, there is an open question as to the privacy and ownership of these data and of the digital twins. Who owns it? So we have clinical data right now, right? We have the electronic health, record systems, and those are protected by federal regulations. And so I think applying these guardrails that are in place to them, and they may have to be extended to digital twins. And I think that'll then dictate, sort of how these digital twins could be used. But the scenario that you mentioned is certainly an interesting, future option. And it could be the people who I know some folks who do clinical trials as a profession, basically, because they pay a lot of money for some studies. And imagine that you can basically sell your digital twin for research purposes. I think that could be very interesting as a additional benefit to population individuals.
That is really interesting. And you're absolutely right. There's a lot of clinical trial participants that are more or less professional clinical trial participants, travel all over the country to participate. So and they can make decent money, actually. Quite decent.
I was surprised that was it, but apparently in some states you can legally participate only in so many clinical trials, and so they would travel to other states.
Well, there's actually a database. There's a nationalized database of clinical trial participants. So, companies like mine, we track that, and we collaborate with other companies because we don't want people on trials or overlap. They have to have enough of a washout from the drug and things like that. But it also enables them to travel around and participate in these trials, which is right now necessary to move drugs forward, until we all have our digital twins that can do the trial for us. So,
Right, exactly. In my over-optimistic mind, there's, if we lived in an altruistic world, right, there's this massive database where anybody who wants to do research has access to digital twins to run these models. But in reality, it's probably, maybe the big pharma companies who run more clinical studies and get access and maybe permission from from clinical trial subjects to kind of own or rent their data, digital twin data, and then they get to run more simulations based on maybe on your technology because they have more data to run from.
I think that's one of the reasons we need to continue to support federally funded, university and institutional research, so this stuff is freely available and doesn't have to be bought and paid for by big companies.
Mm-hmm.
So it's a tradition that we have at BioTalk Unzipped, Tom, that the last question is from our last guest, not knowing who the next person is going to be, something interesting that they want to know about. So this was from Mark Arnold, and he asks, for their personal growth, how do you encourage people to read the literature and present and publish their own work?
That's a great question, and given them, I find that giving our team members the ownership over their projects and enabling them to be the ones who articulate the importance of what they're doing means that if you want to articulate why you're doing what you're doing, you have to go to the literature and understand what others are doing, what are the gaps, and make that connection between the gap that you're solving and what it is that you're doing on a day-to-day basis. And so I think there has to be a purpose for the reading of the papers. Just telling somebody go read papers just to read papers so that you're up to speed on up to date on the science. I don't think that's enough. I don't read papers on a weekly basis just to read papers. I read papers because I need to write a grant, and to write a grant, I need to make a compelling story, a sales pitch to the agency, and to do that, I have to understand what those gaps in the scientific domain are. And so I think to motivate students or scientific staff to read, they need to have that fundamental reason to read, other than just to read. To publish, that's also similar. So we're at a university, and for a student to graduate, they have to publish. Well, technically not, but we make the argument that if you don't publish, you are going to be in a huge disadvantage looking for a job, particularly go into academia, and that is the case. So again, they have that motivation. For other staff, it may or may not be as important to publish, and it really, for me as their mentor and supervisor, I try to understand personal goals of each member, and for those that is to stay in academia, you have to publish. Publications are the currency in academia. In the private sector, maybe not so much. And Chad, you can probably correct me if I'm wrong. But it really depends on what they want to achieve and where they want to go. I think being able to write effectively is critical, and so at minimum you're developing writing skills again, and it's part of communication as well.
Yeah, and it's not, it's not essential to publish, certainly when you're, working in industry, but there is a similar element to that. And I think there's also, when you're coming out of academia, I'll tell you, when I hire people, I like to know that they've had that experience with publication, with the thought that's required, the ability to write, the ability to synthesize your thoughts. And so it is important. And as people want to develop scientifically, you get so caught in your small piece of the process that when you publish, you actually do have to look outside that and assess that literature, and it forces you to think more broadly. So I really, that's certainly, I still publish occasionally, not as much, and not as in depth scientifically as I used to, but every time I do, I'm looking outside of that narrow focus and learning a lot. And just by making that commitment to myself or somebody else that I'm going to publish alongside them, it forces me to do that. And sometimes you need that, motivation to go for it.
Yeah, absolutely.
Yep.
The challenge in academia with I feel like in academia, the sole focus on on the numbers, on how many publications you and what journal you publish in. They are these metrics are a double-edged sword, because we also, some faculty and students, especially early in their careers, they are chasing the numbers because they know that if they're applying for a postdoc position, people are looking at how many papers did you publish and what journals did you
What's
publish.
What's the impact factor? And for sure.
But unfortunately, that's also not the best way to do it, because every time you're writing, so to get a paper published, I think it takes about a year from between the revisions and all of that stuff, and it's the year that you're spending thinking about something that you had done already, instead of working on the next thing and creating something new. And if you do that too much, that is kind of moving backwards rather than forwards, and kind of slowing down the whole process of science.
Interesting. Yeah, sure. Interesting perspective. So, yeah. Well, boy, Tom, I've really enjoyed having this conversation today. These are topics I've wanted to explore with you for the last several years that I've come to know you through the collaborations we talked about earlier, and I could make this go on for 2-3 more hours. We got to meet for a beer when I'm in Nebraska one of these days.
That would be great.
I would love to catch up. So yeah, thank you. I really appreciate it.
Thank you for having me here. Really appreciate it as well.
Yes, thanks for being with us, Tom. And as Chad was saying, I think there's quite a few questions we left unasked just in the interest of time. But it's been great getting to know you, having this conversation, and we'll look forward to kind of getting this out to our audience and out to the world some more. So thank you very much for being with us.
Absolutely. Thank you both.
All right. Thanks, Tom.
Thank you.
Take care, guys.